Good afternoon, everyone. My name is Jordan. Today I will talk about a study on breast cancer cells using bioinformatics and genomic to look at gene fusion event. This research was done on the early estrogen stimulation to see the activity of estrogen receptor beta, which may help fight cancer. In this presentation, I will briefly talk about background and objective of the study, then explain the materials and method used. After that, I will show the main result and finally share the conclusion. Breast cancer is one of the most common cancer and a major public health problem around the world. It's caused by both genetic and epigenetic changes that lead to uncontrolled cell growth. One important genetic event in breast cancer is gene fusion, where two different genes combine and create abnormal proteins that can support cancer development. In breast cancer, estrogen hormone and their receptors, estrogen receptor alpha and beta, play important roles. These estrogen receptors are nuclear receptors expressed by two different genes, ESR1 and ESR2. Generally, estrogen receptor alpha is often linked to a cancer growth, while estrogen receptor beta may act as a tumor suppressor. In 2015, Dago and colleague showed that estrogen receptor beta regulates alternative mRNA splicing after 2 hours of estrogen stimulation, and confirm estrogen receptor as act as a tumor suppressor. In this study, the goal is to better understand if estrogen receptor beta works as a tumor suppressor by limiting the expression of gene fusion linked to breast cancer. In this study, we used existing RNA sequencing data from 2 published studies. The first data was from Dago and colleague, and which includes MCS7 breast cancer cells data with both estrogen receptor alpha and beta. And the second data set was from PNC, and it contained RNA data from healthy breast tissue. All data were downloaded from the NCBI public database and used to perform our bioinformatics analysis. For the bioinformatics part, we follow a simple analysis pipeline. First, we use FASTQ to check the quality of the RNA sequencing data. Then we align the reads to the human reference genome, GRH38, using RNA star. After that, we detected gene fusion event using the star fusion tools. We also assess the gene expression level of our 2 genes, ESR1 and ESR2, using DESEC before detecting gene fusion. We also use RNA software to perform the statistical analysis. FASTQ showed that all reads has a good quality, which means that the sequencing was done correctly. This is shown in the graph on the left, where most of the caps are in the green zone, which is a sign of a high quality. The alignment also was very successful. About 90–80% of the reads match on our genome, as you can see on the table on the left, on the right. We analyzed the expression of our 2 genes, which code for estrogen receptor alpha and estrogen receptor beta. In non-stimulated cells, the gene ESR1 was more highly expressed than the ESR12. After stimulation with estradiol, ESR2 express increased while ESR1 decreased. This switch is clearly shown in the graph. This shows that estrogen receptor beta activated by estradiol can reduce the expression of estrogen receptor alpha. Our analysis of gene fusion and chromosomal interaction show no fusion event in healthy breast tissue, suggesting that gene fusion are specific to cancer cells. In AfSCS breast cancer cells, we detected 30 gene fusion, both with and without estradiol stimulation. The number of genes was similar between the two conditions, and meaning early hormone stimulation did not significantly affect fusion frequency. We also found that about 70% of the gene fusion were intrachromosomal, pointing a local arrangement among the amount and also among the interchromosomal interfusion, chromosome 17 and 20 were the most involved, suggesting the key role in this fusion activity. Interestingly, we found 5 gene fusion that were specific to a non-stimulated cells. After estradiol stimulation, this fusion disappeared, indicating they were repressed. This suggests that estrogen receptor beta may act as a tumor suppressor by switching off harmful gene fusion. Also, 4 out 5 of these gene fusion were interchromosomal and involved chromosome 70. A chromosome often linked to breast cancer progression. So estrogen receptor beta might help protect the cell by stopping long-distance DNA interaction between chromosomes, especially those involved in chromosome 17. Here is a table presents the 30 gene fusion we found in MCS7 breast cancer cells. All of them are recognized as a cancer biomarker, and some are specific to breast cancer. This shows that these genes are not random. They may play a role in cancer biology and might be involved in disease progression or diagnosis. This supports the idea that gene fusion even can help us understand more about how breast cancer develops and how estrogen receptor may help to regulate this activity. To conclude, our findings suggest that estrogen receptor beta may help suppress gene fusion activity in breast cancer cells. Further research is needed to explore how estrogen receptor influences chromosomal interaction and formation of gene fusion. I'd like to sincerely thank my supervisor and the Department of Genetic Biochemistry at Pelleforon University in Cote d'Ivoire, as also many thanks to the Department of Neuroscience, Biological and Movement Science, Section Biology Genetic at the University of Verona, Italia, for their collaboration and support. Thank you for everyone who contributed to this supported this study, and big thanks to the ESB organizers. Thank you. Yeah, we did validation because this study was previously okay. The data of these studies are already be validated, um, and we already published a first article about the study, and one next is coming. No, yes, we did not. we did not do this. And I think this is one of the things we have to do for the future. Okay, generally receptor alpha, alpha beta, estrogen receptor alpha some findings are already show that these receptors are involved in the development of breast cancer. And a lot of big studies have showed the real importance of estrogen receptor beta. That's why we decided to focus our study on estrogen receptor beta and his activity.